Hi, I'm Natalie Davidson, and I'm very happy that the organizers of HiTSeq allowed me to share my work with you today. And I want to start off with a metaphor. So there's many ways that you can bake a cake. So typically you need eggs, butter, sugar, and flour, but say that you don't have sugar that day, you can easily replace it with honey, and you would still end up with a cake. In the same way, you can replace your butter with oil. So in the same way, there's many ways that you can induce an oncogenic event. And in this example, for a MET activation, it's known that it can be caused through through non-synonymous mutation, through alternative promoter usage, fusions, or alternative splicing. And what many pan-cancer analyses have focused on is specifically non-synonymous mutations. And what we want to do in this work is to integrate over many different DNA and RNA level aberrations in order to have a deeper understanding of oncogenic events and to possibly identify new oncogenic events. So this project is done as a large collaborative work through the International Cancer Genome Consortium and specifically PCOG3, which focuses on RNA-seq that has matched whole genome. And this is from many different countries, many different cancer types. So in order to process all of these cancer types to make them comparable, we aligned all of the samples using Top Hat 2 and Star 2, and then we used HiTSeq in order to get the counts, and then they were combined once these two were in good concordance and then normalized using FPKMUQ. So here's a breakdown of all of the samples that we have. So here we have 25 different cancer types, and shown here is the number of cancer types, number of samples within that cancer type, and then shown in the gray bars is the number of matched normals. So here you can see the breakdown of tumor versus normal and the breakdown by sex. So to get more specific of the data that we're going to look at, we have 6 different RNA level aberrations and 2 different DNA level aberrations. And each one of these types of aberrations was done by a group that we are collaborating with through PCOG3. And in order for us to integrate all of these different data types, we have to first realize that they're not all on the gene level. So specifically for non-synonymous mutations, many of these events can occur within a gene. So the first level of summarization that we have to do is make them all somewhat gene-centric view. So for example, for non-synonymous mutations, if there's a 1 in that gene, then, or if the event occurs in the gene, then we give it a 1 or a 0 otherwise. For copy number, we average across the gene, and if the average copy number change is greater than 4, then we give it a 1 because this is a significant copy number event, and a 0 otherwise. And for these real valued gene level aberrations, what we do is within each histotype or cancer type, we z-score them and we take outlying events. And so essentially what we have after all of these summarizations is a binary value for each one of these aberrations, which we then have a vector for each gene sample pair, which we then use to fill this tensor, 3-dimensional tensor, in which we have about 1,000 samples, 8 alterations, and about 16,000 expressed genes. So a reason why this tensor sort of shape is interesting for us is that if we aggregate over the samples, we can do a recurrence analysis. If we aggregate over alteration patterns, we can identify pathway disruptions. And if we aggregate over genes, we can identify specific transcriptomic patterns. So we're going to look at each one of these different types of summarizations. So first, we're going to look at 3 different cancer types in 2 different tissues. So let's focus first on the DNA level aberrations. So in this plot, a point is roughly a sample, and the height of it is the number of genes that have that aberration. So if we look at lymph BNHL, we see that it has many genes with variants, maybe a similar amount that we'd see in kidney RCC. And maybe what you take away from this is that kidney RCC is more similar to lymph BNHL than lymph CLL because they have more similar frequencies of variants. We get a different picture when we look at only the RNA level aberrations. So we see that in splicing, we see much more splicing in the lymphatic cancers than we do in the kidney cancer. So when we have a more holistic view looking across both RNA and DNA levels, we can see a more comprehensive view of each cancer individually. I think this is well summarized in this biplot. So here, each point is a sample, and they're colored by each of their cancer types. So here we see kidney RCC, and over here we see the lymphatic cancers. And we can see that the kidneys down here are negatively correlated to the frequency of splicing. Similarly, we can see this separation between lymph BNHL and lymph CLL because the lymph BNHL seem more correlated with a higher number of fusions, expression outliers, and alternative promoters. So this is looking across and comparing 3 different cancer types. So what can we learn within a single cancer type? So here, this is for our kidney RCC cancer samples. And it's known that the PI3K pathway is recurrently altered and important in kidney RCC. So it's been previously published that missense variants, so these dots, expression outliers, these purple bars, and copy number here are important, or are important alterations in the PI3K pathway. And when we look at these 3 different alteration types, we see that, yes, 37 of our samples do have an alteration in this pathway. But we see that there's many samples that don't have an alteration in this pathway. If we then include the rest of our aberration types, we can see that we can classify or identify 21 more samples that have an aberration within this pathway. So now we're going to take an unbiased view and look at which genes are important. So typically, these sort of recurrence analyses are done on copy number or missense variant frequencies. And the idea of what we want to do is that we want to do this for all of these different alteration types. So let me go through this plot really quick if you haven't seen this. So along the columns are samples, and along the rows are genes. And here we see the percentage of samples altered. And you may notice it's not ranked by the percentage of samples altered. So if you're familiar with these types of plots, that may be strange for you. But we had to do a different scoring metric in order to properly rank these genes. And we did this so we did a second smallest alteration rank. And this is done because it guards against frequent alterations dominating. So for example, fusions occur at a much lower rate than, for example, expression outliers. In order for the more frequent alterations to not dominate, we then transform them to ranks. And then we use the second smallest alteration rank in order to prioritize genes that are heterogeneously altered. So what do we see here? Well, we see a significant enrichment for cancer gene census genes and for PCOG8 identified driver genes. Another thing that we see is that we have the typical players. So we see that we have P10 and TP53 in the top of our list. But also, interestingly, we see genes that haven't previously been identified as either a driver gene or a cancer gene census. So this gene is KLF13, and we see that it has many expression outliers, many splice outliers, and many allele-specific expression outliers. And then when we go into the literature, we see that it's been previously reported that KLF13's overexpression is important for leading to different oncogenic effects. So this also gives us some more evidence that this ranking scheme and that this integrating over different alteration types is showing us genes or prioritizing genes that are important in cancer. So now let's take a look at different pathways across all of the cancers. So here along the rows are different pathways, and along the columns are all of our different cancer types. And here we're only focusing on DNA level alterations, so copy number or non-synonymous mutations. And these circles here are this large light blue circle if it's more than 50% of the samples have an alteration there. And here, these dark blue dots, if it's around half the samples have an alteration in that pathway. And all these other spaces, it's less than half of the samples have an alteration in this pathway. So what we see is that, as expected, the P53 pathways are currently altered across many different cancers on the DNA level. But now if we focus on the RNA level, we see that instead the RTK pathway seems to be most frequently altered. So if we put both DNA and RNA together, so first of all, we see that there's many more pathway alterations, but we get a more comprehensive view on how these pathways are altered. So let's take an example, P53 for cancer samples. We see that it's recurrently altered on the DNA and RNA level. But what we can see is that the proportion of alteration types, so we see that almost 50% of the samples that have an alteration in the P53 pathway, it's due to an RNA alteration, not a DNA alteration. And furthermore, we can have even closer view on exactly how or what those alterations were that altered those pathways. To bring it back to my earlier metaphor, there are many ways in which this pathway could be altered in the same way there are many ways that you can bake a cake. So in conclusion, we performed a comprehensive analysis of transcriptomic alterations across 25 different cancer types. We increased the number of samples found to contain an alteration in cancer-specific pathways, and we identified cancer-relevant genes through occurrence analysis using both DNA and RNA level aberrations. I'd like to thank my lab, specifically Kyung Van Leeman, Andre Collis, and my advisor Gunnar Ratch, and everyone involved in PCOG3, specifically Alvis Brasma and Angela Ann Brooks. Thank you.
Great talk. Do we have questions? Okay, so maybe I can start with the first question. So you showed the approach applied to patients' data. Can it be applied to cell line encyclopedia to see whether some patterns on the RNA level can be associated with sensitivity to certain drugs?
Yeah, this is basically like just giving some credence to the idea that you need to also include, or that's useful to include RNA aberrations. So you can do this on cell line or any sort of way in which you can identify these aberrations. Yeah.
Very nice. Very nice. The integration of these different variations has this question about background frequency, and you elucidated the fact that you're trying to normalize for differences in recurrence frequencies across different data types. So my question is that sufficient, or do you also have other sort of null background complexities, for example, different sequence contexts of different numbers of rearrangements and mutations, and so there might be that a gene is more likely to be recurrently mutated simply because of some local other bias. Is that something you think you need to do, or is...
Okay, yeah, so yes, definitely. So for example, with missense variants, the length of the gene is very important and needs to be taken into account. Specifically for gene length and these other aberrations, we didn't see a specific correlation between there, but it could be that we just haven't specifically looked at all the different types of sequence contexts that would relate to those aberration types.
Hello. Thank you for the nice talk. I was wondering, when you fill your tensor, probably you don't have all the different data types for each sample, so you probably have a lot of missing data. Do you throw those samples out, or do you have a method in working with that?
So no, we ensure that we have that the tensor is completely full, basically, because that's a problem that we didn't want to have to deal with at this time. Yeah.
Okay. Thank you.
Mm-hmm.
It's a very nice talk. I just have one question. Do you think like you can really say that, for example, RNA expression outlier is a real driver. This is really something that you can convince, people, let's say, working in this cancer field to really convince them that it's true.
Well, so for example, if we go to the, I have to go back quite far. So for here, so it's known that gene overexpression is a driver of the PI3K pathway. But for these other alteration types, it's true. We don't know if they're actually driver events or not. And in most cases, a lot of these, you're not sure if they're driver events.
Exactly.
What we hope is that since we very strenuously filter and only take very extreme events, that they may have some sort of functional importance in driving the cancer. But no, we don't have a concrete answer on exactly which ones are functional, which ones are not functional.
Okay, thank you.
You were, nice talk. You were working mostly on the RNA sequencing data, and you applied various tools to detect, for example, indels and so on. But did you try also to confirm this if it were possible from other data types for the same samples, for example, whole genome sequencing or exome sequencing?
Yeah, so the DNA level aberrations are actually done on whole genome sequencing. But we only focus on these two sort of things, whole genome sequence and RNA-seq. We haven't looked into the other ones, but it's definitely possible to look into those, yeah.
So do you have survival data on these to compare whether there's a difference depending on the modality of aberration?
So there is a little bit of survival data, but we haven't looked at making these Kaplan-Meier ones to see if these sort of aberrations have a change in the patient's survivability.
Thank you again, Natalie.